Thank you so much, Robin, for that introduction. Welcome, everybody. Thanks for joining the webinar. Just let Steve take a second and pull up the slides. Thanks, Steve. Today's presentation is FDA's Project Optimus and how will it impact oncology drug development. Our speakers for today are Dr. Claire Dyer and Mr. Steve Sibley. Steve is currently a Vice President of Global Submissions at Certara Synchrogenics. He has more than 30 years of pharmaceutical industry experience, and he's played significant roles in more than 75 regulatory submissions. Joining Steve is Claire, who's currently a Director of Global Submissions at Certara Synchrogenics. Claire has more than 20 years of experience in clinical development and holds a doctorate in adoptive T-cell therapy for oncology. Claire and Steve, welcome to today's event. I'll now turn it over to Steve to start the presentation.
Excellent. Thank you, Suzanne. Welcome, everyone. Good morning, good afternoon, good evening, wherever you're located. Thank you for taking time out of your busy schedule to join us today for this session, where we'll be speaking to FDA's new Project Optimus and how it might impact oncology drug development. So what's driving this? And this is an issue that's been around a long time. And so today, what we want to present to you is how the advent of targeted molecular and biological agents is requiring a change in the standard oncology paradigm for dose selection and dose optimization. And FDA's Project Optimus is really a framework for reforming that dose optimization and dose selection paradigm in oncology. So I'll start us off with some background on exactly what Project Optimus is and looking at the standard oncology paradigm and how that might need to change. And then I'll turn it over to Claire, who will walk us through impacts and implications of the initiation of Project Optimus and with some case studies of potential impacts. And then we'll have time at the end for Q&A. As Suzanne mentioned, feel free to post your questions throughout the presentation, but we'll hold answering those until the end. All right, so what is kind of the purpose of Project Optimus and who's driving this? So FDA's Oncology Center of Excellence is the one driving and creating Project Optimus. The ultimate purpose is reforming dose optimization and selection in oncology. This is being done because the historic cytotoxic agents were designed and were the primary treatment options in oncology. And there's a whole paradigm of study design and drug development that was built around that, but is no longer applicable for these targeted agents. And this issue has been around a long time. I know when I was working in an emerging oncology team in AstraZeneca almost 15 years ago, we were bringing the first targeted agents out of discovery and into first studies in humans, and running into issues with establishing a maximum tolerated dose because the mechanism is so different for those agents than for cytotoxic agents like platins. So FDA's goal here, and what had been all of a variety of discussions from a lot of different organizations and fronts through the years, has led to this Project Optimus because dosing at the kind of the highest level possible up to a maximum tolerated dose can be detrimental in this area, because the treatment duration that may be needed in oncology can be for very long. And if you're using a dose higher than is actually necessary for these targeted agents, you may be increasing the risks of toxicity. So what's the stated goal? So this is directly from the Project Optimus website, and you'll see if you look into this further that FDA has a nice website for this, but no guidance document available yet. So This is still early days for this program. The goal of Project Optimus is to educate, innovate, and collaborate with companies, academia, professional societies, international regulatory authorities, and patients to move forward with a dose finding and dose optimization paradigm across oncology that emphasizes selection of a dose or doses that maximizes not only the efficacy of a drug, but the safety and tolerability as well. There was a very good session organized and led by the Friends of Cancer Research, and we have a link to the document resulting from that that gathering at the end of this slideshow. It was a big part of FDA's development of this Project Optimus effort. So what are some of the specific goals that FDA is hoping to achieve with this effort? So one is communicating expectations for dose finding and dose optimization through guidance, workshops, and other public meetings. So as I mentioned, there's no guidance available yet, but there is definitely intention to get to that point and have guidances related to this. But in the meantime, they're working actively with patients, with investigators, with pharma companies and others to come up with the best strategies for these new agents that are not cytotoxic agents. Second is to provide opportunities for and encourage drug developers to meet with the FDA oncology review divisions early in development before they've embarked on later phase trials. And thirdly, to develop strategies for dose finding and dose optimization that leverages non-clinical and clinical data and dose selection, including randomized evaluations of a range of doses and trials. I'll go through in a moment kind of what that paradigm has been and how different that is than what's being proposed here for Project Optimus. So how did we get here? So historically, the first oncology agents were cytotoxic drugs such as platins, and they've been the mainstay of oncology drug development and treatment for decades. At its core, these drugs kill cells, and they're often doing that in an indiscriminate way. So they're not targeted to a cancer cell or to a particular type of cell. So with this type of dosing, the higher the dose, the greater potential to kill all the cancer cells. However, you're obviously creating toxicity in other parts of the body because you're not specifically targeting only the cancerous cells or the organ or part of the body where the cancer exists. So there's definitely limits to the dose that can be given of these agents before you start causing serious side effects in the patient. So if we look at the overall lexicon paradigm that we're dealing with, and it's really some of the realities of dosing with these cytotoxic drugs, there's a whole field that exists within oncology, and I wanted to just touch on some of these terms here to establish this, because these are some of the terms that there needs to be a kind of paradigm shift in that Claire will speak to in more detail later. So dose limiting toxicities. So this is a side effect that prevents the patient from continuing with the dose, requires them to stop treatment, and so limits the dosing that can be given, and is officially identified as a dose limiting toxicity. The maximum tolerated dose, speak to exactly how that is defined in the phase 1 study in a moment, but this is the maximum dose at which very few if any, patients experience a dose-limiting toxicity. Regimen, so it's not just take a pill each day or take one pill a month or something like that. There's often quite complicated dosing regimens with oncology agents. Could be 100 milligrams intravenously or by infusion once every 3 weeks. And that 3 weeks is defined as one cycle. So you often have these chemotherapy agents given in cycles, and that's what the diagram in the lower right part identifies. Is you give a dose of the chemotherapy agent, you're basically letting that work and letting the body recover for 3 weeks after that before you give the next dose, and then that gets repeated cycle after cycle for the duration of the treatment that's necessary. So that then also leads to terms like drug holidays, dose reductions, and dose interruptions. And any of you who've worked in oncology know how important it is to track and summarize these at the end of a study and pay attention to where these are happening and why. And so a drug holiday is when the toxicity to the patient has reached a point where you don't need to completely stop treatment, but you do need to stop it for some extended period of time and take a break from that therapy and allow levels of whatever was being impacted negatively to return at least towards baseline, if not back to baseline. Dose reductions are perhaps just the first step would be to just reduce the dose to limit the toxicity. Hope you continue to see the same level of efficacy. So in this very simplistic example, it provided maybe you'd go to 50 milligrams every 3 weeks and see how the patient tolerated that dose. A dose interruption, you might just give an extra week or two or skip a cycle to allow levels to return to normal, the patient's condition to return back to baseline before continuing on with the next dose. So as you see, if you've worked in oncology, you're very familiar with these. This is part of the lexicon for anyone who hasn't worked in oncology. These are the things that you have to get up to speed on and get used to seeing in all of the studies and in writing them up and designing them. So if we go to what is that standard phase 1 design, what has it looked like for decades now? And it's this dose escalation to MTD, then with a follow-on expansion phase, either as 1B or 2A. And so how does this work? So once you've defined that starting dose based on animal studies and maybe model-informed drug development, some measure of modeling and simulation of dosing based on that animal data and other inputs, you start with the first giving that dose to 3 patients. If there are no dose-limiting toxicities or only one dose-limiting toxicity, you move on to the next. And if the same occurs there, you move up to the next dose. And depending on the type of treatment, the level of dose escalation will have been pre-specified and how much you jump up from one to the next. But the design is such that you continue moving up this dosing ladder until you hit a dose where 2 or 3 of the 3 patients experience a dose-limiting toxicity. At that point, you step back, usually down 1 dose level, and repeat that. And if you, again, as before with that dose level, do not get 2 out of 3 patients with a DLT, that dose can be declared as the maximum tolerated dose. Now, to some extent, this isn't that different than single ascending dose studies with other therapeutic areas. However, The big difference is that what typically happens with these cytotoxic agents, and has been happening with others, more recent agents in oncology, is that MTD is then worked into a regimen that aligns either as monotherapy or with the desired combination therapy, and carried forward for the remainder of the development program. So that MTD that got established in this phase 1 portion then becomes the dose selected for a dosing regimen that's then tested in a variety of different indications, different tumor types, patient populations, patient subsets, as shown on the right here, or as monotherapy, or all of the above. And then whichever the product shows efficacy in is then moved forward into further through phase 2 into phase 3. And it's quite common for that MTD to just continue as the established dose throughout. So the big paradigm shift is really trying to move from this to more of a dose finding and dose ranging approach that we see outside of oncology with agents in other therapeutic areas, so that you're establishing some confirmation of where you thought you would see therapeutic effect in phase 1, and where limits on toxicity might be now that you've introduced the product into humans. And then looking at multiple doses and potentially multiple regimens in a phase 1b or phase 2a to establish a dose before you carry forward into phase 3. And that's, at the end of the day, what FDA is really looking for, and I think we're all looking for with these targeted agents, where it's not a cytotoxic that just indiscriminately killing cells, and so you're trying to, the higher the dose, the greater the efficacy, but also the greater the toxicity. With these, and as shown in the graph on the right, with these, it's much more like we've all been used to with areas outside of this oncology paradigm. So that 3x3 study design that established the maximum tolerated dose and selection of a single dose or dosing regimen that was then carried forward throughout all the way out to commercial launch, is the goal here is to replace that for these new targeted agents with something closer to a true dose finding, dose selection, not just using the highest dose that's tolerated. So one thing that often happens with these agents is if you put them in that old oncology paradigm, you can never get to an MTD because they're not that cytotoxic. You're more likely to reach a limit on how much can physically be administered, or have tapped out the receptor inhibitor occupancy and blocking that is achievable. And so what's shown here on the right is you see the 50% efficacy level, the ED50, that's reflected here as you move up the curve of response rate and across on exposure. And on the far right, you see where the MTD might sit. But you see that response curve kind of plateaus not too far after that ED50. And so sometimes you might see the red arrow referred to as kind of a therapeutic window, the area between where you've achieved kind of a high level of efficacy and below what might be a maximum tolerated dose. But optimally, what you want to hit is the highest dose you can achieve close to your maximum possible efficacy with the lowest relative toxicity. And so trying to find that balance is where we really want to get to. rather than just going straight to the MTD, which you can see on this depiction, is generating a lot more toxicity than necessary with very little, if any, increase in efficacy and benefit. All right, so with that, I will hand over to Claire, who will get into more of kind of the impacts and implications of Project Optimus. Claire?
Thanks, Steve. Would I be able to have my video started, please? Thank you. So thanks, Steve. So before we were thinking about Project Optimus, as Steve has said, the maximum tolerated dose was the phase 1 standard for oncology studies. And it was rare for the phase 2 or 3 studies in oncology drugs to evaluate more than one dose. However, outside of oncology, most drugs are evaluated in randomized dose ranging trials that support a broader understanding of the impact of different doses on efficacy and toxicity. So the maximum tolerated dose, as Steve has said, is a dose initially developed for the systemic chemotherapies. And obviously things have moved on. And so now we are finding that there are limitations to the MTD paradigm. So there may be limitations in the evaluation of the interpatient variability. So the tolerability versus benefit is not necessarily going to be the same for each patient. And we need to drill down on what patients potentially are going to tolerate a drug better and benefit from a higher dose versus those patients who are going to tolerate a drug a little bit less better than others, but will benefit actually from a lower dose. So dose response or exposure response has been quite ill-defined in oncology studies. During the trials, we generally, as Steve has said, there have been dose reductions are pretty common. So if you're using the same dose throughout your studies, when you're getting to your phase 3 registration studies, you are going to see a lot of those dose reductions, dose holidays, and this is not something that is really required or really wanted within your phase 3 study. So, but conversely, dose increases are not that common. So you're not actually picking up those patients who could probably have a higher dose of your drug, and they would benefit from higher exposure because that's not how the studies are set up. You have this one dose going through. And as Steve has also said, that the MTD is not necessarily the optimal biologic dose. Obviously, you could pour in a lot of these biologics, but actually they would be saturated at a lower dose with a target receptor, and that's very likely, almost always, to be lower than the maximum tolerated dose. So that we have a major impediment to the selection of optimized dose in oncology because we don't have enough data coming out of the clinical studies from multiple dose levels in the phase 1, 2, and 3 studies. And can you go to the next slide? Sorry, Steve. But there's going to be a few people who have, there's perceived challenges to Project Optimus, and these are laid out by the Friends of the Cancer Research annual meeting last year. The main thoughts were that dose finding studies would be too time consuming within the oncology space and prevent patients from quickly getting the drugs that they need. Obviously, this is oncology studies are different from other therapeutic areas in the way that we want to be getting these drugs out to patients very quickly. But the dose finding studies were perceived to be too time consuming. Also, lower doses of drugs are not as effective as higher doses is commonly thought from the point of view of key stakeholders who are used to working with chemotherapy agents. So the continued focus on identifying and using the MTD may be driven by a desire for kind of speed and some misconceptions in the scientific and pharmaceutical community. There's obviously this notion that it's not worth performing randomized dose finding clinical trials because they're very time consuming, may delay drug development and keep these life-changing therapies away from patients. And it's also thought because of the higher dose leads to higher efficacy is born out of the chemotherapy drugs, but it's also in light of the fact that we generally know that cancer patients will actually tolerate side effects. We are, you get cancer, you tolerate as much chemotherapy as you can to ensure you've got the best chance at remission. It's a common thought, and it's obviously born out of the chemotherapy, the drugs. But we still think very much on these terms. And even after spending 20 years working on biologics, I still thought in these terms when a friend of mine recently was being treated with a chemotherapy that, oh, brilliant, she's in a high dose, they're going to throw everything they can at her and she's going to recover. And actually, that's the notion that we need to get our head around if we're looking at drugs that are other than chemotherapy drugs. So we need to challenge these paradigms that we're thinking about. So essentially, we need to start by changing our way in thinking. We need to rethink that dose finding studies are important to understand the therapeutic window of the drug, and that's required to treat a range of patients effectively and minimize toxicity. If we plan these studies properly, they can be used to inform the registrational study of the drug, the phase 3 study, and should be done pre-registration if done in a timely manner. And knowing a true therapeutic window and a true benefit of the drug will mean ultimately patients will be treated more appropriately, they're likely to miss fewer treatments, remain on treatment for longer, and a more appropriate dose may also provide an opportunity for patients that we wouldn't have otherwise treated to have access to a drug. So this is the goal. The goal of the dose finding studies is to adequately characterize the exposure safety relationship as well as the exposure efficacy relationship, sometimes called dose safety, dose efficacy, to select a dose with a suitable benefit risk profile, which is an acceptable level of toxicity, but also of maximize your efficacy, that's going to be brought through into the registrational trial and ultimately used post-approval if you get approval of your drug. So as Steve alluded to in his slides at the beginning, the shift in thought from the MTD dose to the dose finding studies as proposed by Project Optimus centers around education being very key to this. Sponsors do need to understand the utility of appropriate dose finding trials. And in the long term, these will support further analysis of their products and ultimately ensure that the greatest number of patients benefit from the product. So some of the educational points that the FDA have proposed is the enhanced guidance from the FDA, which obviously we haven't seen yet, but the engagement with sponsors in selecting doses for oncology registrational trials. It's really important that we also don't miss out the patients, and the patient-informed consent documentation needs to really communicate reasoning why they may be on a lower dose and the rationale for why it was chosen based on the data, based on modeling, which ultimately inform the activity and the reason why it's been chosen in that particular clinical trial. Sponsors are going to be and should be encouraged to publish results from their dose finding studies. The more we know, the more we learn. And we need to educate providers and patients about the outcomes from dose finding studies. This could also support understanding of how lower doses do not always necessarily lead to lower efficacy. We need to get out of that thought. So next slide, please. So in addition for us to kind of changing thought processes, the point of Project Optimus is also to provide a strategy, like a fundamental change in approach. And for those of you who work out of the oncology space, this is not reinventing the wheel. This is often what we see with the biologics in other therapeutic areas. So what should we be doing in oncology, according to Project Optimus, is we should start obviously at the preclinical data stage. That's no different to what we've seen before. but it's leveraging the preclinical data to define the target saturation points and exposure effectively, which narrows the range of doses that we can use in phase 1 studies. It's really helpful to be able to identify at the preclinical stage biomarkers that can translate from the animal models or protein modeling into the clinical trial design at the early phase. And it helps to characterize the PK, the pharmacokinetics, and the pharmacodynamics metrics in addition to those that you would use generally for patient selection. So if you're kind of familiar with the ECTD structure, so when you're doing a submission, this is the kind of data that we find in our non-clinical modules, but it's also a crossover that we find in our clinical modules because it's really pertinent to the labeling, to what you're going to be telling the prescribers down the line. So we need to be very considered as we progress into further drug development with very informed decisions coming out of preclinical data. Well-defined biomarkers at this stage that we can actually use in the phase 1 studies really can support the understanding of dose-response relationship along with your safety and efficacy data. So we can have biomarkers that measure activity, so efficacy, and can be used to track the relationship between plasma exposure and change in tumor endpoints, for example. And safety is always can be tracked through biomarkers, for example, neutrophil counts, your general labs that you would see coming up in early phase studies. So the proposed pathway is that the phase 1 trials, the phase 1 studies should include adequate PK sampling here. So what we're talking about is intense a sampling regimen. So the patients will be sampled at intervals where we can see what the curve is doing from your drug, from your biologic. And this enables a clear determination of all the PK properties of the drug. And it's a really preliminary characterization of your dose exposure relationships. And when it is feasible, and when you have gleaned this information out of your non-clinical data, pharmacodynamic endpoints can be incorporated into these early studies to determine drug exposure that results in, for example, inhibition of the drug target. And then if we move into phase 2, these should go really beyond the assessment of drug activity and can include adaptive designs or exploration of doses in a randomized manner. And we should be continuing sparse PK sampling at the very least, because that adds to the knowledge, so you can gain a sense of the relationship between the exposure and the clinical outcomes that you're going to be introducing into phase 2 to a larger extent than phase 1. But if possible, in phase 2, you can keep the measurements of the pharmacodynamic endpoints, because then you have an addition to that data that you have already collected in phase 1. And after the completion of some randomized dose finding studies, data from these trials can be starting to be analyzed, modeled to characterize exposure response relationships. So for both efficacy and safety, and then be integrated with any PK or PD analysis results. And this informs the final dose for a registration trial. And this is not going to be, in these studies for biologics, the maximum tolerated dose. So if there's a clear difference in the benefit with an acceptable safety profile compared with a lower dose of a high dose, then the higher dose can be selected for the registration or the phase 3 study. But if there's efficacy and safety are similar between a lower dose and a higher doses, and you have modeled that for your phase 3, the lower dose can be selected as the final dose for the registrational trials. And in phase 3, you're incorporating, pop PK sampling in order to further evaluate the relationship between covariates, for example, influencing exposure. And pop PK modeling and exposure response for efficacy is for the key clinical outcomes and safety. Will inform the exposure differences due to intrinsic and extrinsic factors. So you can already start seeing that you are going to be informing your label if you get your product licensed. And it doesn't really end there. Post-marketing, you can still monitor with the therapeutic drug in a post-marketing setting to enable the dose for an individual patient to be adjusted. Down the bottom of the slide, I've actually mentioned inclusion of patient-reported outcomes, just because I didn't want to miss it off. It's particularly important in oncology because it's really common with cancer and the treatments with cancer, and there's psychosocial factors which will affect subjective experiencing and functioning. So if you can incorporate patient-reported outcomes within your phase 2, phase 3 studies, these can also be they can also be linked with the exposure that you're getting out of the PK measurements. So the proposed approach that, as I've just presented, is a collection of a maximal collection of exposure data and data regarding tolerability across a range of doses. And it can be used to define the threshold of exposure needed for both your anti-tumor effect, your efficacy, and determine peak exposure that correlates with excess toxicity. So you don't want a point that you don't want to hit above for your dose. And obviously, as I've said, the collection of drug exposure and tolerability data, as well as all this ongoing evaluation of adverse events and dose modifications from patients in real-world settings, is also useful for post-marketing evidence generation. So with that in mind, what is the optimal timing to include these dose comparison studies? I've already alluded to the fact that we need to get in early with phase 1. But regarding discussions with regulatory authorities, it is advised that discussions with the FDA regarding dose finding studies, you should be talking about these as early as possible. And we're talking pre-IND setting, which is pretty early. And sponsors really need to start considering additional meetings to discuss dose optimization. Obviously, there's going to be, you have your end of phase 2 meeting with the FDA, but there are opportunities when needed to be discussing dose optimization throughout the drug development. So ideally, randomized dose comparison studies and exposure studies would be performed in the pre-marketing setting, because this allows review at the submission and with the maximum amount of information on exposure response that will probably lead to, and hopefully what you hope to lead to, is a positive submission. So the ideal time to be performing randomized dose comparison studies is pre-marketing setting, because such studies can potentially improve the chance of approval, minimize the chance of excessive toxicity that is taken through to your phase 3 study, and minimize the inadequate efficacy that can be observed because dose studies is too low. If we do these studies within the pre-marketing setting, the argument is that it builds a foundation for the scientific rationale for choosing dose and supports any further updates to drug approval. So for example, if you go for a monotherapy with your biologic, but you have a wealth of exposure response data, it may aid the approval of other therapies, such as combination therapies with your drug, or changes in route of administration. You may be experiencing, fewer clinical holds or fewer add-on studies, which cost time and money in later development. And I'm not saying that the approach of doing this pre-marketing setting is without challenges. Obviously, dose comparison studies in the pre-market setting do have some challenges. That's why there is, there's going to be pushback on these kind of these dose response studies. The thinking that they may slow the development of new cancer drugs, or maybe a bit burdensome if there's some uncertainty as to whether you will ultimately get your drug approved. But actually, post-marketing setting, if you leave these kind of studies that the FDA are going to want in the future until post-marketing, then that comes with its own sets of challenges. The ones I could think of were, that many people don't want to enroll into studies where there's a drug already on the market, or they may be, trials outside of the US where there is less enforcement from the FDA, and commitments often can't be met, or they're rarely completed within timeframes. So what do these dose finding studies ultimately, what do the FDA want these dose finding studies to look like? Well, they want them to be randomized with at least 2 doses. And the dose finding studies are not necessarily going to be powered for statistical comparison across doses, but it is important that it's of sufficient size so that you understand the general dose exposure response relationship. That's for safety and for efficacy, including the minimally active dose. So there's selection of doses is advised that are pharmacokinetically distinguishable, so don't have overlapping PK exposures. And the lowest dose is the minimal dose expected to provide activity on PK/PD analyses. And the highest dose is selected to ascertain whether the dose increases as a result of increased activity with acceptable toxicity. And the selection of the initial doses for the dose finding studies, as well as methods for determining which dose to move into the registrational trials, are going to be influenced by the therapeutic properties and the patient population being studied, or combination versus monotherapy, regimens. And following trials, the analysis should be done on exposure response, which informs a registration trial, which is what I was talking about on the phased slide earlier. Ooh. So I have an example here. I wanted to provide an example of what it really looks like. Nivolumab is a checkpoint inhibitor, so it's an antibody that binds PD-1, if I remember correctly, and it's therefore a biologic that is not going to benefit from a maximum tolerated dose approach. And it is very successfully been licensed in I would call a plethora of studies. So there is, I think there's about 11 indications on nivolumab, and this is how the early dose studies panned out. So there was initial anti-tumor activity and safety data from a large phase 1b study performed, and it was open label but dose escalation study at doses ranging from 0.1 to 10 mg per kg, and it was done in a regimen every 2 weeks, and there was 306 patients. And this was a bucket study, advanced malignancies, sometimes called an all-comers study. So initial study had both intensive and sparse PK data sampling, and there were a lot of different advanced malignancies included in the protocol. And the phase 3 dosing regimen was selected based on integrated analysis of safety and efficacy across the tumor types and dose level and dose response relationships of efficacy and safety, and pharmacodynamic biomarkers from the phase 1b study. And they even used early tumor shrinkage as a surrogate marker of survival in the context of dose selection. So that was something that they did a pharmacodynamic approach on early on. And then there were further dose expansion cohorts enrolled for various cancer types. So we've got melanoma here that's got, I think, 5 doses, non-small cell lung cancer, which has got 3 doses, and RCC, which has got 2 doses. But the patients across the tumor types in the phase 3 were enrolled into cohorts of 3 to 6 per dose level, and dose escalation proceeded when a minimum of 3 patients had completed the safety evaluation period. So we're kind of seeing a shift here into a more a dose finding studies that are more acceptable to find a therapeutic range for patients, so we can move on to treating more patients. And also, as you can see, that the amount of different indications that they have got out of these initial dose expansion cohorts and then moving on to phase 3 within different cancer types has been a very successful submission strategy for them. So I'm going to hand you back over to Steve to conclude.
Excellent. Thank you. Thank you, Claire. So a few take-home messages to review here at the end before we close. So the maximum tolerated dose paradigm is no longer acceptable, both from standpoint of FDA is now looking very hard at that and giving it a lot of scrutiny, but also it's really an appropriate paradigm for a cytotoxic agent, but not for these new molecularly targeted therapies. FDA's Oncology Center of Excellence is the one initiating and driving Project Optimus. And this will affect not just new programs, so studies, so products that are just coming into first-in-human programs, but also FDA is looking at ongoing programs and the doses being used in those currently. Model-informed drug development, other modeling approaches can help tremendously in terms of informing that initial trial design and dose selection, both for the first-in-human studies as well as for any dose-ranging dose-finding studies as you move into phase 2. There is a major emphasis and focus on education and agreeing on new approaches to dose selection. FDA is pushing that. You saw it was one of their specific goals was to have a lot of discussion on how best to address this. And in that regard, for your specific program, for your product, the recommendation to meet early and needed with FDA to discuss your approach and how to proceed. So with that, I would like to thank you all very much. I will turn it over to Suzanne to review some of the reference materials we've linked to at the end here and facilitate the Q&A.
Fantastic. Thanks, Steve. Thanks, Claire. We'll looks like we've got some questions from the audience, so we'll try and take as many as we can in the remaining time, and then we'll wrap up with a few short announcements. So, Steve and Claire, one of our attendees would like to know if you have any advice on how to apply these recommendations to cell therapy products.
Yeah, I don't know if, Claire, you may have some specific insights on cell therapy.
Yeah, I have started working on a couple of cell therapy submissions, and it is effectively going, the sponsors are trying to go down the same route as for the biologics. Obviously, you know, you're looking at cell numbers rather than a dose of an antibody, for example. But they have performed a dose finding or a dose increase study with different cells, but with it's very much within a range. And obviously the one problem with the cell therapies is if you're generating cells from a person to put back into a person, there are all the technical problems in getting that dose back into a person. So whilst they try to do fundamentally a dose ranging study, what they actually effectively need to do is get as many cells back into that person as possible and overcome the technical difficulties with, you know, doing that. So it hasn't been the dose study hasn't been so set up as you would do with like a kind of another biologic, like a monoclonal antibody or similar. But there is they're trying very hard to get a rationale for the dose out, which is going to be based on a lot of PK modeling based on the number of cells that are persisting within the patients. So there's going to be an element of the modeling behind it in order to inform the dose, and we're relying very heavily on what's coming out of modeling.
Great, thank you, Claire. So the issue of dose selection and dose optimization isn't a new concept. So do you think that, why do you think that the FDA is has issued Project Optimus now?
That's a good question. I think I don't think there was any single event that triggered this. And yeah, as I mentioned, the issue is absolutely not new. These agents have been around for 15 years or more, and so you could make the case that this should have started 15 years ago. But I think so. I would just say it's high time. It's time that we stop treating all of these as, with a cytotoxic MTD approach, and looked at a more rational kind of dose optimization approach for these, as Claire especially spoken to. So yeah, I don't know of a specific event. I know that Friends of Cancer Research meeting was if you had to point to a trigger, that might be one. And it's worth reading their summary minutes from that. It's very good accounting of all of the considerations, especially from the patient perspective.
Do you have any guidance on monotherapy versus combination therapy dose escalation? So how would you recommend properly selecting a combination starting dose?
Yeah, Claire, you want to take a stab at that one?
Yeah, so depending on, so if you have done your dose escalation studies coming out of early phase, it may be that you only have a dose for your monotherapy. And from what I've seen is that when we have a combination with like a biologic with a non-biologic drug, a cytotoxic drug, generally the dose is a reduced dose with the cytotoxic, but there has to be borne out of you are going to have to provide some information from earlier studies with the combination. So there can be studies in like phase 2-3 studies where you do more than one dose within that area. So you might be looking at a high dose, low dose with combo arm versus mono, or versus mono plus placebo arm.
Awesome, thanks, Claire. Somebody wants to know about how Project Optimus could be implemented for rare disease indications in oncology, because the small number of patients could limit the feasibility of dose exploration.
Yeah, it's a good question. I think yes, there are absolutely limits on the dose exploration options because of the limited number of patients. I think you could make the case that it's even more important to ensure you're getting to the right dose with those patients. So I would think this just puts a little more pressure on getting that initial starting dose right, where you come out into this your initial trial. I don't know, Claire, if there's anything you would want to add to that.
Yeah, I just think maybe so I've worked on an orphan drug with a biologic where it was incredibly helpful to get in with the FDA at that pre-IND stage to discuss the dose with relevant people, because actually with orphan drug, where there's very little patients, where there's vanishingly little known about it, it was a case of trying to get the experts in at an early stage. And also within, I think I've seen cases where there's been like phase 1 studies where they have potentially identified PD biomarkers, but have performed studies in healthy volunteers. Obviously, we don't have that in the cancer space if we're trying to go straight in with the cancer, but is possible.
Great. Somebody has a question about other health authorities. Are they considering the same approach or a requirement from FDA? So for example, are the EMA or the PMD likely to accept a development program that follows the dose optimization that's described in Project Optimus?
Yeah, I think it's a good question. I think there's two parts to that. One is I'm not aware of any Project Optimus type program or guidance from EMA or PMDA or others. However, the idea here is that you are actually selecting a much better dose from an efficacy and safety, benefit risk standpoint. So if you've done that additional work on identifying that dose, then I can't imagine running into an issue with a health authority in another country, other than you'd still want to be having those same discussions with them and explaining what you're doing and explaining why you haven't just gone to an MTD approach. So I think the same guidance of speaking with them early on and running your development program by them is definitely valuable. But at the end of the day, you should have all the justification from your trial results and data to demonstrate that the dose is safe and effective.
Yeah, it looks like we have time for one last question. And if there's any questions that we're not able to get to, we can always follow up with folks by email. So Project Optimus, as you know, is a initiative of the FDA's CDER division, the Center for Drug Evaluation and Research. Do you have the impression that CBER, the Center for Biologics Evaluation and Research, is adopting Project Optimus to the same extent as CDER?
I'm not positive about. I know it's coming out of the Oncology Center for Excellence specifically. And there's certainly been discussion already of biologics in the same breath as small molecules. But in terms of CDER versus CBER, I don't have any insights. I don't know if Claire, you have any.
No, I'm not aware of CBER having an initiative. I mean, being as other therapeutic areas, we're pretty much, oncology space is going to be catching up with them regarding the dose finding anyway. I'm not sure whether there's other initiatives in other parts of the FDA.
Great. Well, thank you so much, Steve and Claire, for that really informative presentation. We just have a few announcements before wrapping up. So we'd like to let folks know on the call that next week, Certara's Dr. Julie Bullock will be presenting an e-chalk talk with AAPS on how to prepare for Project Optimus. So if you want to learn even more about this topic, you can go to aaps.com and find out about it there. Also, to let you know that on May 3rd and May 5th, the FDA is partnering with ASCO on a dose optimization workshop. And we've got some several events in the past that on this topic that you may want to check out. Rap is going to share the slides from this presentation with everyone, so you'll have this information at your fingertips. Next slide, please. We'd also like to share some additional resources on relevant publications, FDA guidance documents, and thought leadership on this important topic. On behalf of Certara, I'd like to thank our speakers, Steve and Claire, for an excellent presentation. And we'd like to thank RAPS for allowing us to host this event with them, and thank our audience for their attention and their excellent questions. This concludes the webinar. Goodbye and have a great day.